Welcome to MitoCast by Mito Foundation. We share information and resources to support people with Mito, their families, and the wider community. All content is created by Mito Foundation. AI text-to-voice technology is used only for the production to make our resources easier for everyone to access.
Today, we're talking about genetics. It might sound like a big, complicated topic, but if we start at the very beginning and build step by step, it becomes much easier to understand.
Understanding the basics of genetics is important because changes in our genes can sometimes cause conditions like mitochondrial disease, which we call Mito for short. So where do we start?
Let's start with the body itself. Did you know your body's made up of about 40 trillion cells?
40 trillion? That's hard to even picture.
It is, and cells are the basic building blocks of life. They don't work on their own, though. Similar cells team up to make tissues, like muscle or nerve tissue. Those tissues then form organs, like your heart or your brain, and organs don't work in isolation either. They link up in body systems, such as the nervous system or the digestive system.
So cells really are the starting point for everything in your body.
Exactly. And if we were to look inside a cell, it would be a bit like peeking into a tiny house, filled with different structures, each doing its own job.
Those structures inside the cell are called organelles, right?
Yes, organelles are like the cell's organs. Each one has a specific job. Most are busy with things like turning food into energy, moving materials around, or cleaning up waste.
And some of them carry DNA too, don't they?
That's true. The nucleus and mitochondria are two special organelles that have their own DNA.
Shall we start with where DNA fits in?
Let's do that. Inside those organelles is DNA, short for deoxyribonucleic acid. DNA is like your body's instruction manual, which guides how you look and how your body works. It's written in just 4 letters, A, G, C, and T. These letters pair up like the rungs of a ladder, and the order they're in is what gives your body its set of instructions.
Okay, but what are genes?
Genes are sections of DNA, like chapters in that instruction book. You inherit them from your parents, and together they make you unique.
But what do genes actually do?
Genes contain instructions to make proteins. Proteins are the building blocks and tools of the body. There are about 20,000 different proteins. Some build muscle, skin, and organs. Others act like tiny machines that carry messages or speed up chemical reactions. The structure and function of your body are controlled by different types of proteins created within the cell.
And where do chromosomes fit in?
Good question. Inside the nucleus, DNA is packaged into structures called chromosomes. You can think of them as books on the library shelf, each one full of recipes. Humans usually have 46 chromosomes, arranged as 23 pairs, one set from each parent. Each chromosome holds many genes, and together they make up your unique set of instructions.
So let me check I've got this straight. The body is made of cells. Inside cells are organelles. The nucleus and mitochondria are special because they carry DNA. DNA is the code. Genes are the chapters, chromosomes are the books, and proteins are what those recipes make. They keep the body running.
That's a good way to put it. Now we can zoom in on the 2 places where genes are stored: the nucleus and the mitochondria.
Shall we start with the nucleus?
Great idea. The nucleus is the control center of the cell. Almost all your genes are stored there. We call them nuclear DNA.
And how can nuclear DNA changes cause mito?
Out of the 20 to 25,000 genes in nuclear DNA, about 1,100 are there to help the mitochondria do their job.
And if one of those genes changes, the instructions can come out wrong?
Exactly. The protein it's meant to build might be faulty, or it might not be made at all. And without the right proteins, the mitochondria can't make energy. the way they should.
So nuclear DNA changes can cause mito by messing up the proteins that mitochondria rely on.
That's right. When those proteins don't work properly, the mitochondria can't produce enough energy, and that's what leads to mito.
And what exactly are mitochondria?
Mitochondria are the energy centers of the cell. They turn food and oxygen into the fuel your body needs. And most cells don't just have one. They can have hundreds, or even thousands, depending on how much energy that cell needs.
Wow, so the more energy a cell uses, the more mitochondria it has?
Exactly. And here's another interesting part. Mitochondria carry their own small set of genes called mitochondrial DNA. Each mitochondrion has 37 genes.
So how do mitochondrial DNA changes cause mito?
Those 37 genes give instructions for proteins directly involved in making energy. If a change affects one of those genes, the energy-making process breaks down. The result is not enough fuel, especially for high-energy organs like the heart, brain, and muscles.
So if I put this together, nuclear DNA changes stop mitochondria from getting the proteins they need, while mitochondrial DNA changes directly break the energy-making process.
That's right, and because either type of DNA can be involved, mito, can be passed down in different ways. These different ways in which DNA is passed down is what we call an inheritance pattern.
So depending on which DNA is affected, the inheritance pattern changes?
Yes, that's right.
Shall we walk through the inheritance patterns?
Sure. With nuclear DNA, a change can be dominant or recessive. A dominant change shows its effect if only one copy of the gene has changed. A recessive change usually shows only if both copies are changed, one from each parent.
So someone might carry one recessive change and not have symptoms. They are called carriers of the condition because they are not affected by it. But if their partner carries the same change, their child could be affected.
That's true. Then there are X-linked changes, which sit on the X chromosome. Females have 2 X chromosomes, while males have 1 X and 1 Y. Because males only have 1 X chromosome, they're more likely to be affected if that X carries a change. Females with 2 X's often have a backup copy, though they can still be affected in some cases.
And mitochondrial DNA changes, they always come from the mother, right?
Yes. A mother's mitochondrial DNA is passed to her children because mitochondria are inherited only from the egg.
But sperm have mitochondria too, don't they?
They do. The mitochondria in sperm are packed in the tail to power the swim. After fertilization, those mitochondria are broken down. The embryo keeps only the mother's mitochondria.
So the father's mitochondrial DNA isn't passed on to the child?
Yes, that's right. That's why mitochondrial DNA is inherited only from mothers.
And even then, not all children are affected in the same way.
That's correct, and it's a really important point because of something called heteroplasmy. Heteroplasmy is when not every mitochondrion in a cell carries the same version of DNA. Each of your cells can contain several thousand copies of mitochondrial DNA. Some copies may carry the change, while others don't.
So it's really about the mix?
Correct. The proportion of changed to unchanged mitochondria can differ between people, and even between organs in the same person. That's why symptoms can vary so much.
And I've heard sometimes genetic changes aren't inherited at all.
That's right. Some changes appear on their own when a child first begins to develop. These are called de novo changes. De novo just means new. You might also hear them called sporadic changes or mutations. All these words mean the same thing, a change that wasn't in the parent's DNA, but shows up for the first time in the child.
So if I recap, nuclear DNA changes can be dominant, recessive, and/or X-linked. Mitochondrial DNA changes come from the mother, but sometimes they can also appear on their own, and symptoms vary because of heteroplasmy,
That's exactly right.
which leads us to genetic testing. Shall we dive in?
Sure. Genetic testing usually starts with a blood test. From that, scientists can look closely at your DNA. Think of it like a careful proofread of your body's instruction book. It reads the DNA, letters A, G, C, and T, and checks for changes. Many of these differences are harmless, but some can cause conditions like mito.
And when scientists do this proofreading, are they looking at all the DNA or just part of it?
Great question. Sometimes they look at the whole genome. That's the complete set of all your DNA instructions, like having every recipe ever written down. Other times they focus just on the exome, which is the section of DNA that codes for proteins. That's only about 2% of the genome, but it's the part scientists understand best, and where many known disease-causing changes are found.
And in Australia, testing usually needs a specialist, right?
Yeah. Testing for mito is usually ordered by a specialist. You can ask your specialist about genetic testing or get a referral to a clinical genetics service. If the test meets set criteria, Medicare can cover the cost. In fact, both whole genome and whole exome sequencing are subsidised in Australia.
So, what kind of sample is used?
Often blood. Sometimes a tissue sample, such as muscle, is better, depending on the test and what the health professional is looking for.
And there are different kinds of tests. Can we run through them?
Yes. Let's start with the bigger ones. Whole genome sequencing, or WGS. This is the most complete test. It reads every single letter of your DNA, all 3 billion of them. It covers both nuclear DNA and, if requested, mitochondrial DNA. It's like reading every recipe in every book, cover to cover, even the ones no one looks at very often. Because it's so broad, it can pick up all sorts of changes, from single letters swapped around to whole pages added or missing.
So, whole genome sequencing is the most thorough?
Exactly. It gives a huge amount of information, but the challenge is working out what all that information means. Sometimes it finds a change, but scientists don't yet know whether that change actually causes mito. So the results can be complex. Still, whole genome sequencing often simplifies the diagnostic process because it checks everything at once.
So, whole genome sequencing is like the biggest, broadest sweep, reading every page to catch anything that might matter.
Exactly.
What about whole exome sequencing, or WES?
Whole exome sequencing looks at just a small slice of your nuclear DNA, about 2%. This is the part that gives the instructions for making proteins, and we call those sections exons. Think of them as the key steps in a recipe, the part that actually makes the meal. Most known disease-causing changes are found here, so whole exome sequencing zooms in on the part of the DNA where the best clues usually are.
So, whole exome sequencing doesn't read the whole genome, just the protein coding parts?
That's right. Which makes it cheaper, faster, and easier to analyze than whole genome sequencing. And because many genetic conditions, including mito, are caused by changes in these coding regions, whole exome sequencing is often a really useful tool. It's also offered by more Australian labs than whole genome sequencing.
So, whole exome sequencing is more focused. It zooms in on the key parts we know the most about.
Yes, that's right.
And the third one, mitochondrial genome sequencing.
Yes, this one focuses only on the mitochondrial DNA. Remember, each mitochondrion has 37 genes. This test reads through all of them carefully. It's like pulling one book off the shelf, the mitochondrial DNA book, and proofreading every line.
When would that be used?
It's especially useful if someone has clear symptoms that suggest a mitochondrial DNA change. It can confirm those changes directly. But because whole exome sequencing doesn't always pick up mitochondrial DNA changes? Well, mitochondrial sequencing is often combined with whole exome sequencing. That way, you're checking both nuclear DNA and mitochondrial DNA together.
So this one is more like a fine-tooth comb just for the mitochondria.
That's a good way to put it.
So to recap the main ones, whole genome sequencing is the broadest, reading everything. So nuclear and mitochondrial DNA. Whole exome sequencing looks at just the protein coding parts, which is where most known conditions are found. And mitochondrial genome sequencing checks only the mitochondrial DNA and is often paired with whole exome sequencing.
Spot on. And then there are smaller, more targeted tests. Targeted panel tests look only at a specific group of genes. For example, if someone's symptoms point strongly to a condition like MELAS or LON, the panel can quickly check just the genes most often linked to those. The results are usually faster and sometimes cheaper, which can help families get answers quicker. But because panels cover a small number of genes, maybe 20 or so, they can miss changes in other genes. That's why if a panel doesn't find anything, a doctor might recommend whole genome sequencing or whole exome sequencing next.
And what about if a change has already been found in a family? How do doctors test other relatives?
That's where cascade testing comes in. It's used when a genetic change has already been found in a family. Instead of starting from scratch, doctors can test other relatives specifically for that one known change. It's faster, cheaper, and often covered by Medicare. Cascade testing helps show who else in the family carries the change, even if they don't have symptoms yet.
So targeted panels are like a quick, focused check when there's a strong suspicion. And cascade testing is like following a known change through a family.
Yes, panels are useful for quick answers when the signs point to a specific condition, and cascade testing is the fastest way to see who else in a family carries a known change.
So to sum it up, whole genome sequencing and whole exome sequencing give the widest view. Mitochondrial sequencing zooms in on mitochondrial DNA. Panel tests are a quicker option when symptoms suggest a certain condition. And cascade testing traces a known change through a family.
That's it.
What about reproductive testing? Is that an option?
Yes. There are a few kinds. Carrier testing is for people with a family history of mito. If a relative is known to have a genetic change linked to mito, doctors can test you specifically for that change. This type of testing is often free through a public hospital genetics service. Carrier screening is broader. It's usually done before pregnancy and looks at many genes, sometimes hundreds. It checks whether future parents carry changes that could be passed on to their children. Some carrier screening includes certain genes linked to mito, but not all. Screening is often arranged privately and may not always be free. Then there's prenatal testing, which can check a baby's genes during pregnancy. And during IVF, there's pre-implantation testing, where embryos are checked before being implanted. These options can help reduce the chance of passing mito onto children.
And what about those genetic kits you see advertised online?
They're not recommended for mito. Many are done overseas and aren't held to Australian clinical standards. Results can be incomplete or confusing. For mito, it's best to work with health professionals who can ensure accurate testing and clear explanations.
So testing can be broad or targeted depending on the situation.
And before looking at the benefits, it's important to think about a few things.
Like what?
Well, people often worry about life insurance. At the moment, in Australia, insurers can use genetic test results when deciding cover and costs. That might mean higher premiums, or sometimes being refused cover altogether. Because of this, some people choose to organise their insurance before going ahead with genetic testing.
That makes sense. And what about the personal side of it?
Yes. There's also the emotional impact. Learning that you or someone in your family carries a genetic change can stir up feelings like worry, guilt, or sadness. Some people also feel angry, anxious, or even depressed. On the other hand, some feel relief from finally having answers. Everyone responds differently, and support can make a big difference in adjusting to what the results mean.
Are there practical things to think about too?
Definitely. Cost and eligibility can play a part. Some tests are funded through Medicare or hospitals, but not all. Others may bring out-of-pocket costs, depending on the type of test and where it's done. Public hospital patients often pay nothing, while private testing may involve costs.
And doctors often ask about family history too, don't they?
That's right. Before a test, doctors or counselors may ask about relatives with things like early deafness, blindness, or seizures. Drawing up a family tree helps them spot patterns and work out risks for different family members.
So it's not just a medical step, it's emotional, financial, and practical too.
Exactly. That's why genetic counseling helps. Genetic counselors are university-trained health professionals who provide both clear information and support. They explain what testing could mean for you, talk through possible results, support decision-making, and discuss options if you're thinking about future children.
And who else might be part of the genetics team?
Alongside counselors, there are clinical geneticists, doctors who specialize in diagnosing and managing genetic conditions. Sometimes social workers are also involved to help support families with the emotional and social impacts. Together, they form a team around the person being tested.
So once someone has thought about those things, what are the benefits of genetic testing?
There are several. First, testing can confirm a diagnosis with more certainty than clinical signs alone. For many people, having a clear answer reduces the stress of uncertainty and the constant search for explanations.
That makes sense.
Genetic testing can also speed up the process. Without it, families may go through multiple tests and appointments over months or years. A genetic test can sometimes give an answer much faster.
And sometimes it can avoid some of the more invasive tests, like muscle biopsies.
Exactly. That means less stress and fewer hospital visits for many people. Testing also helps with proactive management. If doctors know the exact genetic change, they can plan monitoring and care earlier. It doesn't mean they can predict every detail, but it allows them to watch for known risks and sometimes step in sooner with support.
So it can help families prepare.
Yes. It can also help other family members. If a change is found, relatives can choose to be tested too. That way, they can understand their own risks, even before they show symptoms.
And that ties into reproductive planning.
It does. For parents who want children, knowing about a genetic change means they can explore options, like IVF with pre-implantation testing or prenatal testing. It helps them make informed choices about the future.
And you mentioned earlier that testing can open doors to research too.
Yes. Right now, there are over 100 clinical trials worldwide looking at treatments for mito, including gene therapy. Most of these trials require a confirmed genetic diagnosis before someone can join, so testing can create opportunities to access new treatments as they're developed. Another benefit is ruling out mito. Research shows that some people with mito-like symptoms actually have a different condition. Genetic testing can help identify that, so they can get the right treatment.
The benefits range from certainty to faster answers, better management, family planning, and even access to research.
That's right.
And where are these tests done in Australia?
Two major services are worth mentioning. The Victorian Clinical Genetics Service offers a range of genetic tests for mito, including whole genome sequencing. Many of their tests are bulk billed under Medicare, which means no out-of-pocket cost for those who are eligible. pathology, Queensland also offers testing. Currently, they focus more on targeted mitochondrial DNA testing rather than broader sequencing options. Each service is a little different in what it provides, so your specialist will guide you to the right option.
And how long does it usually take to get results back?
It varies. Often it takes around 3 to 4 months. In urgent situations, say if results could change immediate medical care or pregnancy planning, it can be faster. In some research projects, urgent testing can even be completed in a matter of days.
Sometimes I hear results can take longer too, can't they?
Yes, especially if the test finds a genetic change. But it's not clear whether that change causes mito. DNA contains a huge amount of information, and interpreting it isn't always straightforward. Some results come back uncertain. Over time, as research provides more clues, those results can be reclassified.
So even an uncertain result today could become clearer in the future.
Exactly.
Once results come back, what do they actually mean?
The report will explain whether a genetic change was found and whether that change is likely to cause mito. Some results show a clear pathogenic change, which means the gene isn't working properly and it can cause disease. Other results may show a benign change, which is harmless.
And sometimes the result is uncertain.
Yes, that means a change was found, but scientists don't yet know if it causes mito. DNA holds so much information, and we're still learning what all of it means. Over time, research may show whether that change is important. In that case, the result can be reclassified.
So if someone gets an uncertain result, they should keep in touch with their doctor or genetics team.
Absolutely. And that's where genetic counselling really helps. Counsellors explain what results mean, talk through risks for family members, and support people in making decisions.
And we mentioned earlier that counselling also helps with planning for children.
That's true. Counsellors can explain reproductive options. They tailor the conversation to each family's situation, so people feel informed and supported.
So if I step back and look at the big picture, our bodies are made of cells. Cells have organelles, and two of those, the nucleus and the mitochondria, carry DNA. DNA is the code of life, arranged into genes and packed into chromosomes. Genes make proteins, and proteins keep the body running.
That's right. Mito happens when changes in nuclear or mitochondrial DNA stop mitochondria from making enough energy. These changes can be inherited in different ways, or they can appear spontaneously.
And genetic testing helps identify those changes. It comes in different forms, involves practical and emotional considerations, but can also bring huge benefits, from answers and planning to family options and access to research and clinical trials.
Yes, that is an important point. Lots of clinical trials require a genetic diagnosis. If you'd like to explore more, all the information we've talked about today comes from the Mito Foundation's resources. We've linked them in the show notes, and you can also find them anytime at mito.org.au by searching genetics and Mito.
And of course, you can always call our helpline if you'd like support. You've been listening to MitoCast by Mito Foundation. To learn more or find support, visit mito.org.au or call our helpline on 1300977180. Make sure you follow MitoCast wherever you listen to this podcast to help us spread the word by sharing it with others impacted by Mito. Thanks for listening.